Okay, so hopefully we're watching this video after we watched part one about transcription. This is part two of protein synthesis. Once our mature mRNA transcript has left the nuclear port's now going to go find a ribosome. We're going to begin the process of translation. So the big picture review, let's take a look at what's going on here with the overall big picture and what we're trying to do. This is the central dogma of molecular biology here. This is the idea that we have a genetic code that's contained within our cells, and we have within every single one of our cells, with the exceptions of something like the red blood cells, which doesn't contain a nucleus, we have genes, and all of our genes inside of every single one of our cells. What we need to do is target a specific gene for production. That process is called protein synthesis. When we say production, what we mean is the product is eventually going to be a protein. In transcription, we looked at DNA going to RNA using RNA polymerase. That's called a transcript or transcription. RNA then needs to be moved into the form of a protein. That's not saying this molecule gets turned into the protein, but rather it's read and then the protein is assembled. So here's a summary of what we're going to be looking at in translation, where we're going from the mature mRNA transcript to proteins. A fundamentally different molecule with different pieces. So in the first phase here, we're going through initiation, which the ribosome is the thing that's responsible for both synthesizing and reading the mRNA transcript and synthesizing the protein. A ribosome is going to attach to the initiation sequence, or what we're going to call the start codon, which is for us universally AUG, or methionine is the amino acid that AUG codes for, but it's going to be the start codon. Part two is the elongation, where transfer RNA molecules are going to bring amino acids that correspond with the mRNA codon transcripts. And then termination is going to happen where a stop codon is read and the strand is cleaved off, and eventually we're going to get a protein out of that. Okay, so in this first phase of initiation, what I'd like to have you look at here is really just a diagram. So what we've got is the ribosome with its two subunits. And then what we've got here is the start codon. So here is initiation, which AUG, the start codon, is going to be read at the P site by the ribosome. From there, what's happening is this tRNA molecule, this twisted bit of RNA that has three bases sticking down at the bottom of it, is going to match up. It's going to be complementary with the codon, the three-letter code on the mRNA transcript, AUG. Notice how U, A, and C are complement to the AUG start. Along with that, it's going to bring this amino acid, which we're representing here as a circle labeled MET, which stands for methionine. What we have is this recognition happening here at the P site, in which the A site is going to be where the anticodon goes in and starts recognizing the codon. So here we can see CCG is the next one. So what should match up? GGC, and here we see the anticodon GGC. For this transfer RNA, what it's bringing along with it is the amino acid proline. So proline will be next. So this is how we begin to synthesize a protein. This is going to be our protein, our polypeptide chain. This is the mRNA. They're involved with each other, but they're not the same thing. This doesn't magically get turned into a protein, so let's keep that in mind. Here's methionine. We got the anticodon GGC coming in. And then here, what we're going to have is a shift of the ribosome down, in which in this site, the P site, what we're going to have is methionine joining with the proline, and now we have methionine, and it's going to be covalently bonded with the proline next to it. They formed a covalent bond through dehydration synthesis. They got rid of a water molecule there between the amine group and the carboxylic acid group. And now they're joined together. So now we're starting to form a chain, and that's the process of elongation. And this is going to continue until we reach a termination sequence. So here we're going to shift down. You can see proline and methionine are growing. Here comes a tyrosine molecule. It's going to start coming in. So these things are starting to really link together. And then an alanine will be next until eventually we get to this region downstream, which is a stop codon, which is going to tell the whole process to stop. And for the mRNA transcript, you're done, and the polypeptide will break off. So when it hits that termination sequence and it is cut off, it's going to move to a special protein called a chaperonin in order to be folded into different shapes. That's eventually going to form a protein, and ultimately that came from the DNA code. Now we're far away in the process from being involved with DNA, but ultimately it started with DNA. Three stop codons can stop the process, UAA, UAG, or UGA. When those are read, we know that we're ready to fall off, and we have a polypeptide chain. Okay, From there, we get these different levels of structure. So what we just made was the primary structure of a polypeptide, where here we've got the carboxylic acid group, over here is the amine group, the ends, the C terminus and the N terminus. And here what we've got is the primary structure, where it's literally just the amino acids all in a row. The secondary structure begins to happen when we've got interactions between the carboxylic acid groups and the amine groups, in which we get things right here. You can see there's an interaction where we're getting these typical patterns called a beta pleated sheet, or we also get these interactions due to hydrogen bonding, in which we get an alpha helix. And these are all due to the carboxylic acid and the amine groups interacting with each other. From there, the R groups, which are the unique parts of amino acids, will start interacting. When they start interacting, you get even more folding. So this is the initial folding. We're going to get even more folding when the R groups start interacting, because some of them are ionic and they're going to be attracted to other ionic groups, or some are nonpolar that will be attracted to other nonpolar groups. So you get this tertiary form when the R groups start interacting. Then when you get multiple bits folded together, when you get multiple tertiary polypeptides folding together, what you eventually get is the quaternary structure, or what we've got here is a mature protein. That's a very specific shape that can go do a very specific job. Maybe its job is to be a protein kinase that's going to pass a phosphate group and a phosphorylation cascade and cell signaling. Maybe it's going to be a receptor protein out in the membrane of a particular cell. Maybe it's going to be RNA polymerase that's going to help with the process in the first place. They have hundreds of thousands of jobs. And they all do one very specifically. They do one very, very well. And they ultimately get made through this process of protein synthesis, transcription, and translation put together. So that's it, guys. Your jobs here are to know about what translation is, the role of the ribosome in this process, what the transfer RNA molecules do, and how they bring amino acids with them. That peptide bonds are going to be the bonds that are formed in between the amino acids in the growing polypeptide chain due to dehydration synthesis. That covalent bond we just call peptide bond when it's in a polypeptide chain, that they will eventually fold into proteins. Okay, I hope that's helpful. That wraps up the review that I have on protein synthesis. If you have any questions, be sure to see me during advisement or any other time, and I'll be sure to answer those questions for you guys. And good luck.